Hi everyone. Today we are going to talk about how Sanger sequencing could be applied for plant genetic improvement that leads to improved yield and quality, tolerance to unfavorable environmental conditions, and resistance to disease. Let me introduce standard Sanger sequence to you. Standard Sanger sequence serves as a basis for modern and increasingly automated technique. As the first generation sequencing technique, Sanger sequencing was invented by Frederick Sanger in the year of 1975. In terms of DNA sequencing, Sanger sequencing is regarded as the gold standard. The human genome process was sequenced using Sanger sequencing in the year of 1977. Sanger sequencing is known as the chain termination method. It consists in using one of the strands from double-stranded DNA as template to be sequenced. This sequencing is made by chemically modified nucleotide called dd-oxynucleotide (DNTP). These DNTP are marked for each DNA bases by DDG, DDA, DDT, and DDC. Once they are incorporated into DNA strand, they prevent the further elongation and the elongation is complete. Then, we obtain DNA fragment ended by a DNTP with different size. The fragments are separated according to their size using gel, where the resultant band corresponding to DNA fragment can be visualized by an imaging system such as X-ray and UV light. For single and low throughput DNA sequencing, Sanger sequencing has been widely utilized for 3 decades and remains used nowadays.
Sanger sequencing workflow consists of three major steps. The first major step is the chain termination PCR of the DNA sample. The reagent that needed in this step is ddNTP, primer, DNA sample, dNTP, and DNA polymerase enzyme. ddNTP is the important reagent in Sanger sequence because they lack of OH group, causes DNA synthesis to terminate when they are incorporated into the growing DNA strand. DNA sample will go through the process denaturate, annealing, and elongate. DNA polymerase extend the strand until it randomly added a furan labeled ddNTP, which terminate the DNA strand. Multiple of the different length of the fragment DNA will produce through few cycle of chain termination PCR. Polyacrylamide caprylase gel electrophoresis is the second major step. The DNA sample first loading into wells. The power is turned on and the DNA fragment migrate toward the positive electrode because the DNA is negative charge. The fragments are separated by size and then the smaller fragments move faster. A scanning laser and fluorescence detector are used to detect the position of DNA fragment. The last step is gel analysis and determination of the DNA sequence. The electrophoresis result translate into the chromatogram that can be visual using the DNA analysis software. The color of ddNTP labeled with a different fluorescent dye that can detected by fluorescence detector. The A is green, D is red, G is black, and then the C is blue. The reading of the DNA sequencing is based on the peak of the color, and then the DNA bases is generated from 5' to 3'.
In plants, Sanger sequencing has contributed to quantitative trait loci mapping, which aims to identify the loci of genes causing variation in complex quantitative traits. This loci identification method helps in pinpointing the specific genes as well as their associated function. For instance, genes which encode for desirable traits such as disease resistance, high nutritional value, and increased crop yield could be determined and combined with other favorable alleles forming allied lines. Thus, by knowing how QTR responds under various genetic conditions allows the development of superior crop types through conventional breeding or cultivation. Besides, it is also possible to directly introduce the favorable genes to a variety of species through transgenic approaches. In addition, the Sanger sequencing method is widely applied in the identification of mutations such as point mutation and frameshift mutation, splicing, and even single nucleotide variants. This knowledge is especially useful in understanding the genetic basis of diseases in crops, thereby producing resistant varieties by genomic engineering or selective breeding. During the addition of a ddNTP to the extending sequence, the base will glow in response to the corresponding nucleotide. In sequences where heterozygous variants are found, two fluorescent dyes of the same intensity will bind to the particular loci, whereas the anticipated dye color will be entirely replaced by the color of the new base pair during the presence of a homozygous variant. Furthermore, due to its ability to identify DNA nucleotide sequences, the Sanger sequencing method also contributes to the understanding of genetic diversity, therefore, making it possible for researchers to compare and analyze the nucleotide sequences from a wide range of plant species or populations. This information gathered could then be subsequently aid in the production of high quality crops that are resistant to pests and tolerant to diverse environmental stresses, thus enhancing yield stability.
When a gene responsible for a desired trait, such as disease resistance and environmental tolerance, are identified by Sanger sequencing method, it enables the discovery of the molecular markers that closely related to these desired genes to select specific traits in crop breeding. Marker assisted selection is the technique that utilizes molecular markers to select the trait of interest. So researchers use them as sign or flags to detect the presence of desired genes in offsprings from crossing of parent plant carrying the desired gene with other parent plant that does not carry the desired gene. So upon selection, plants that carrying the molecular marker related to the desired gene are sent for further breeding. Molecular markers can be used for early generation selection and improve the accuracy and productivity of plant breeding compared to conventional breeding. Besides, Sanger sequencing has the capability to accurately sequence specific regions in the plant genome, thereby assisting in gene editing or gene modification. Plant genome editing or modification technology provides a powerful tool to precisely modify plant genomes for crop improvement. So Sanger sequencing is commonly used to validate CRISPR-Cas9 gene editing workflows, ensure successful gene editing. So this ensures the development of crops with enhanced traits such as disease resistance, tolerance to environmental stress, and improved yield.
The first challenge is large size and complexity of plant genomes. This is because plant genome is characterized by significant size and complexity which can be attributed to the various processes such as gene duplication, alternation gene spacing, and polyploidy. These processes contribute to the large size and complexity of plant genome, posing a challenges for comprehensive sequencing using Sanger sequencing methods. Next, low quality sequences within the first 15 to 40 bases due to the primer binding. This makes it harder to ensure that primer are binding correctly and generating high quality sequence from the start in plant genome. Despite the advancement of next generation sequencing technology, single sequencing remains a valuable and important tool in plant genomics. Single sequencing serves as gold standard method for validating and detecting the single nucleotide variants or sequences obtained from the next generation sequencing platforms. Besides, the combination technique of single sequencing and next generation sequencing allow for an improvement of their respective strengths. Signal sequencing is a helpful tool for completing and filling the gaps in the next generation sequencing assemblies.